Hello friends, today we are going to learn about screening and strain improvement. We have already learned about screening, its types, objectives and strategies. We have also learned somewhat about strain improvement. We know the term mutation and its types. So today we'll begin with mutagenesis, which is a process by which genetic information of an organism is altered in a stable manner. Mutagenesis occurs in nature spontaneously and sometimes due to exposure to certain agents known as mutagens. So mutagens are physical or chemical agents which causes mutation in a DNA. Mutagens are chosen on the basis of their simplicity, safety and availability and simplicity of usage. There are certain agents which are known as the physical agent and can cause mutation in a DNA. And a very good example is ultraviolet light. Ultraviolet light actually causes two adjacent thymers to base pair among themselves in a strand of DNA, which results in a distortion of the DNA strand. The disadvantage of ultraviolet light is that it is absorbed by glass and it is also not effective in colored organisms. Next we move on to chemical agents. There are various chemical agents which can act as mutagen and they can be categorized into two types. Number one, which affect non-replicating DNA, for example nitrous oxide or hydroxylamine, and others which affect replicating DNA, and the examples are base analogues or alkylating agents. Next we move on to types of mutation. Mutation can be classified into the following types according to the change it is creating into the DNA. Number one is point mutation, addition, deletion, and frame shift mutation. Now, what are point mutations? Point mutations are actually small but significant changes which often occur in a single nucleotide base. And deletions are removal of information from the gene. Deletion can range from a single nucleotide to an entire gene. And additions are addition of extra nucleotides into the gene. Now, the frame shift mutations are either addition or deletion of nucleotides into the gene. When this type of mutation occurs, the reading frame of the gene gets shifted. Now, here is the example of a sentence which is written as, "The man saw the dog running," which is a normal sentence with a normal meaning. Now, if the letter G get altered and T is replaced here, then the sentence gets changed and the meaning of the sentence is also changing. This is an example of point mutation. And in the next sentence, if the entire word dog is removed, then also the meaning of the sentence is changed. And this is an example of deletion. Now, in the next sentence, if a letter S is added into the word dog, then also the sentence is becoming that the man saw the dogs running. This also causing a change in the meaning of the sentence. And this is an example of addition. In the last sentence also, if the letter D is replaced from the word dog and it is inserted in the previous word the, then also the frame of the reading is getting changed. This is an example of frame shift mutation. This figure shows a point mutation in the DNA which causing an alteration in the corresponding mRNA. And the figure below shows a deletion of nucleotide in the DNA which is again altering the corresponding mRNA. In this figure also there is an addition of an extra nucleotide in the DNA which is causing an alteration in the mRNA. And here there is a substitution of this nucleotide which is again changing the mRNA and incorporation of a wrong amino acid into the protein. Next we come to types of recombination. There are various types of recombination, and recombination is a process by which a DNA is inserted into an organism. These are the types of recombination. that is recombination in bacteria, recombination in fungi, protoplast fusion, transposons, and recombination by the use of recombinant DNA technology, which is in short known as rDT. Now we come to recombination in bacteria. There are various ways by which recombination in bacteria occurs, and transformation is one of them. Transformation is a process by which foreign DNA is absorbed into a cell and the DNA then get integrated into the recipient genome. And this figure shows how transformation occurs. A recipient cell absorbs certain DNA fragments, and the DNA fragment which is complementary to the recipient genome get integrated into it and gets recombined. The other DNA fragments are then degraded and the cells then become transformed. All cells cannot show transformations, and the cells which show transformations are known as competent cells. Next we move on to conjugation. Conjugation is a process of DNA transfer from one cell to another via cell-to-cell contact. A donor cell having the F plasmid transfer a copy of it into the recipient cell, and then both the cell become F plus cell. This is how conjugation occurs. Next is the process of transduction. Transduction is a process by which a bacterial DNA gets transferred into another bacteria by the means of bacteriophage. This method is a well-established research tool in case of bacteria, but have a limited usage in fungi. The picture shows that when a bacteriophage infects a bacterial cell, the DNA is get inserted into the bacterial cell. The phage then produces certain proteins which digest the bacterial DNA, and the phage DNA is then replicated and all the necessary protein for the phage particles are then produced. After that, the phage particles are packaged. But during this process, mistakenly some phage particles take up the host DNA. And when these phage particles again transduce another or infect another bacterial cell, and if the bacterial cell from the phage gets a complementary sequence in the DNA, it gets inserted into the recipient DNA. Thus, the cell becomes a transduced cell. Next is recombination in fungi. There are various methods by which recombination in fungi occurs, and parasexual recombination is one of them. This type of recombination is found in certain fungi where sexual cycles have not been observed, for example in Penicillium and Aspergillus. This figure shows how parasexual recombination occurs. Next is protoplast fusion. Protoplasts are formed from bacteria, fungi, yeast or actinomycetes when dividing cells are caused to lose their cell walls. And the fusion of protoplast is greatly enhanced by the usage of polyethylene glycol. This picture shows that two protoplasts A and B fuses together having both the nucleus and both the cytoplasm. Then they forms a hybrid of both the cells. Next we move on to transposons. Transposons are actually DNA sequences that can change its position within the genome and sometimes results in duplication. Mutagenesis by transposons has a lot of advantages. In this picture, this transposon is getting cut out and then it is inserted into a new location inside another gene, which results in the disruption of the gene. Next, recombinant DNA technology. It's a process which enables isolation of genes from an organism. It also enables the amplification, study, altered, and the insertion of the DNA into the organism. This is a very simple picture which depicts that some parts of the DNA can be erased and it can be redrawn. according to the interest. This is a picture which shows how rDT occurs. It's a picture of a plasmid which have two resistant gene. One is for ampicillin, one is for tetracycline. If the tetracycline resistant gene is disrupted and the DNA of interest is inserted inside this and the plasmid is then transformed into bacterial cell, then the bacterial cell will grow into the medium where tetracycline is not present but ampicillin is present. That is, it will show sensitivity to tetracycline and resistivity to ampicillin. Now, the components of rDT. There are mainly three components for rDT. Number one is source of DNA, that is the DNA of interest. Number two is the vector. Vectors are artificial or natural plasmids where the DNA of interest is inserted. And number three are the host. Host are the organisms. They can be prokaryotic or eukaryotic where the vector and the source of DNA are transformed. Now, the source of the DNA can be a DNA fragment digested with restriction enzyme, or it can be a complementary DNA, or sometimes it may be synthetic DNA. Vectors can be plasmids, cosmids, or bacteriophage, and hosts can be prokaryotic cells as well as eukaryotic cells. Now we come to plasmids. Now what are plasmids? They are actually small, circular, dispensable genetic elements. They have their own replication origin and can replicate autonomously inside the host cell. This is a picture of a plasmid PBR322 which has two resistant gene. One is for ampicillin, another one is for tetracycline. If the DNA of interest is inserted in any one of this gene, then the bacteria will show resistance for the other gene and will not show the resistance for the gene where donor DNA is inserted, and this will help in selection. Next we move on to phages and cosmids. Phages are actually viruses of bacteria consisting of a molecule of DNA or RNA and a protein code. They bind to receptors on bacterial cell and transfer their genetic material inside the cell for reproduction. And cosmids are artificial vectors made up of plasmid and phages. Cosmids can replicate like plasmid because they have their own origin for replication at a high copy number. Now we come to complementary DNA, in short known as cDNA. A complementary DNA is a DNA which is synthesized from a messenger RNA template in a reaction catalyzed by the enzyme reverse transcriptase and DNA polymerase. This process shows how cDNA is formed in rDT. First, mRNA from a cell is isolated, and then by the help of the enzyme reverse transcriptase, cDNA from the mRNA is made. Then the DNA, that is the cDNA, is inserted into the plasmid. The plasmid are then transformed into bacterial cell and the bacterial cells are allowed to grow. The DNA can then again be isolated and sequenced. Next we come to formation of DNA. How cDNA is formed? cDNA is actually formed from mRNA having a poly A tail. A poly DT primer is attached with the poly A linker of the mRNA. Then the enzyme reverse transcriptase synthesizes the cDNA from this linker complementary to the mRNA. This is the cDNA, having a hairpin loop at the 3' end. Then with the help of alkali, mRNA is removed. Then from this flanking region from 3' end, DNA polymerase I synthesizes the double strand of the cDNA. Nucleases are then used to degrade the RNA primer and the hairpin loop, which results into a double-stranded linear cDNA. Next we come to selection of strains. After doing all these mutations and recombinations and rDT, One is required to select the strain of interest. And selection of strains is done by two methods. One is random, another one is rational. In random method, after inducing the mutations, the survivor from the populations are randomly picked and then they are tested for the ability to produce the metabolite of interest. And in the rational process, the selection is done on the basis of the specific traits of the cell. Next we come to replica plating, which is a type of rational selection, and this picture shows how replica plating is done. Replica is done from a master plate to another plate containing a minimal medium of the colonies grown in the master plate. Now, after incubation, if some of the colonies in the replica plate are not found which was originally present in the master plate, then these colonies are thought to contain the plasmid of the interest, and these are known as the nutritional mutant. Now, the conclusion of the topic. Screening and strain improvement are the two very important parts of industrial microbiology. Strain improvement is essential to increase the production of the desired product, and the recombinant DNA approach has succeeded in introducing new genetic material into a convenient host microorganism and amplifying genetic material. Thank you.